Students, in my last class I have mentioned about the glycolytic pathway. Now, in that pathway I have mentioned that how glucose is converted to pyruvic acid, and that process I have already mentioned that process mainly takes place in the cytoplasmic fluid of the cell. And I have also mentioned that one molecule of glucose is producing 2 molecules of pyruvic acid, and pyruvate is the end product of the glycolytic pathway. Now, if we see this picture, then we can find that this is the glycolytic pathway and pyruvate is the end product. Now, this particular reactions are taking place in the cytoplasmic fluid of the cell, and now this is the end product. Now, this product is to be utilized in very many purposes. Now, under anaerobic condition, this pyruvate may get converted to ethanol and carbon dioxide. It may convert it to lactate, and this pyruvic acid, when it further going for this respiratory chain and entering to the respiratory chain from the cytoplasmic fluid, that it is converted to acetyl CoA, and this acetyl CoA is being utilized for respiratory process. And for this conversion of pyruvate to acetyl CoA, a molecule of carbon dioxide is being released. Now, when it is entering to the respiratory chain, it is this particular process is taking place in the mitochondria of the cell. Now, it has to be transported from cytoplasm to this mitochondria, and the first product being 6 carbon compound citric acid. This particular cycle is otherwise known as citric acid cycle or the tricarboxylic acid cycle, and as this particular cycle has been discovered by the scientist Krebs, this particular cycle is also known as Krebs cycle. Now, whatever may be the name, whether it is citric acid cycle or TCA cycle or Krebs cycle, it matters that end product of glycolytic process, that is the pyruvic acid, is converted to acetyl CoA, and then this acetyl CoA is participating in this particular reaction. Now, this acetate in the form of acetyl CoA is derived from pyruvic acid and other metabolites and is oxidized to carbon dioxide in the citric acid cycle. Now, the 3 stages of cellular respirations are this acetyl CoA production, that is from glucose, fatty acid and amino acids. So, this acetyl CoA is playing an important role in the respiratory chain of living cell. In the stage 2, acetyl CoA is being oxidized and it starts the reactions for TCA cycle, yielding the reduced electron carrier. Now, when these electron carriers are being produced, when the cycle gets complete, then it enters to the electron transport chain and oxidative phosphorylation. That means, how this chain is getting completed? First, glucose to pyruvate, not only this glucose, the source is from glucose to pyruvate, from beta acid oxidation also acetyl CoA can be formed. It can also form from amino acids. So, whatever may be the source, this acetyl CoA production is also another important factor. Then acetyl CoA is getting oxidized and then it starts the TCA cycle, and when this TCA cycle is completed, then electron transport chain and oxidative phosphorylation starts, and it ends with the this production of ATP, and that way it completes the respiratory process. So, the citric acid cycle at a glance, if we see, the citric acid cycle involves the conversion of carbohydrate, fats and amino acid into carbon dioxide and water. In aerobic organisms, citric acid cycle makes up the final stage of catabolism when acetyl CoA is completely oxidized to carbon dioxide. This is also known as this Krebs cycle or TCA cycle, as I have already mentioned. It is a central integrated pathway that harvests the chemical energy from the biological fuel in the form of electrons in NADH and FADH2. NADH and FADH2 then transfer the electron via electron transport chain to the final electron acceptor, that is oxygen, to form the water, and here the total respiratory cellular respiration is getting completed. One high energy compound is produced for each cycle of citric acid or TCA. The electron from TCA cycle are made available to the electron transport chain in the form of 3 NADH and 1 FADH2, and ultimately the energy is provided for oxidative phosphorylation. Now, when we will be completing TCA cycle, we will just see that how many molecules of NADH and FADH2 are produced along with some byproducts. The citric acid cycle is controlled to all respiratory oxidation, oxidizing acetyl CoA from glucose, lipid and protein catabolism in aerobic respiration to maximize the energy gain. Now, when we will be completing this TCA cycle, we will also see that how enriched a particular cell is when it completes this particular cycle. The cycle is this cycle also supplies some precursors for biosynthesis. All enzymes are in the mitochondrial matrix or inner mitochondrial membrane. That means, the glycolytic process though takes place in cytoplasmic fluid, but in mitochondria this TCA cycle takes place. Now, it is very important to know the entry of pyruvate into the TCA cycle. Pyruvate is formed in the cytoplasm as a product of glycolysis. For entry to TCA cycle, it has to be converted to acetyl CoA. Oxidation of pyruvate to acetyl CoA is catalyzed by the pyruvate dehydrogenase enzyme complex, which is present in the mitochondria of the cell. Mitochondria consist of inner and outer membrane. Also, the matrix of this mitochondria, where most of the enzymes are present except the succinate dehydrogenase, which is present on the inner membrane of mitochondria. Now, what we have learned, that pyruvate is to be converted to acetyl CoA, and then acetyl CoA is entering to this mitochondria, and inside this mitochondria the pyruvate dehydrogenase enzyme is present. Now, we have to have this particular that enzyme. What we are talking about is the pyruvate dehydrogenase, and this particular enzyme is playing a very important role in conversion of pyruvate to acetyl CoA. Now, if we see this pyruvate dehydrogenase enzyme, it is a very complex enzyme. Now, if we see its structure, we will find that it has got 20-sided polyhedron structure, and if we see actually the core moieties of this pyruvate dehydrogenase, then we can find that the core of this particular enzyme is made up of dihydrolipoyl acetylase. So, this is the enzyme, it is the core enzyme of pyruvate dehydrogenase. Now, here this enzyme has got 60 identical peptide chain. So, you can understand that how complex this core moieties are, and its molecular weight is approximately 3.1 million Dalton. So, you can understand that how complex this particular molecule is. Each contain covalently linked lipoic acid. So, this is a huge enzyme, and it is obviously allosteric in nature, intracellular, and it plays a very significant role as far as the TCA cycle is concerned. Now, here if we see this pyruvate dehydrogenase enzyme, it has got the core moieties, which is dihydrolipoyl transacetylase enzyme, to which that pyruvate dehydrogenase of 20 such molecules, this another enzyme pyruvate dehydrogenase has 20 such molecules. Each molecule has the molecular weight of 1,54,000 Dalton is attached to this. This is the core moieties. To this core moieties, that pyruvate dehydrogenase, which of 20 such molecules are attached, each are having 1,54,000 Dalton, and to this particular moiety another enzyme called dihydrolipoyl dehydrogenase is also attached, which is 5 to 6 molecules of 1,10,000 Dalton. So, you can understand that how complex this pyruvate dehydrogenase molecule is. So, this is the core moieties. That means, we can understand that pyruvate dehydrogenase is present in the mitochondria, which mainly composed of 3 main enzymes, the core being the dihydrolipoyl transacetylase, where 60 identical peptide chains are present in these core moieties, to which 20 molecules of pyruvate dehydrogenase is also attached to this, and 5 to 6 molecules of dihydrolipoyl dehydrogenase enzyme is attached to this. Now, here this is all about this core structure, but besides this, to this pyruvate dehydrogenase enzyme, 5 to 6 molecules of pyruvate dehydrogenase kinase is also attached, of which are of molecular weight of 62,000 Dalton and 7 molecules of pyruvate dehydrogenase phosphatase enzyme is present of molecular weight of 1 lakh Dalton. So, you can understand that though it has got 3 particular enzymes and these 3 enzymes have direct role in conversion of pyruvate to acetyl CoA, but these last 2 enzymes pyruvate dehydrogenase kinase and pyruvate dehydrogenase phosphatase, these 2 enzymes are playing a significant role in the regulatory process of TCA cycle. Now, we will come to this particular complex structure and we will also see the role of these 2 enzymes as far as the regulation of TCA cycle is concerned. Now, as I have already mentioned that this pyruvic acid can be produced from glycogen to glucose to pyruvate, lactate to pyruvate, fatty acid, and this pyruvate is amino acid to pyruvate and this pyruvate is getting converted to acetyl CoA. Fatty acid can also give acetyl CoA and this acetyl CoA is entering to the TCA cycle. Now, acetyl CoA, if we see its structure, it has got one acetyl group and to which the CoA structure is attached to this. So, this is the structure of acetyl CoA. Now, if we see the conversion of pyruvate to acetyl CoA, then we can find, as we have already learned, that pyruvate dehydrogenase complex is playing a significant role. So, here we can see that this pyruvate in presence of this NAD and coenzyme A is producing acetyl CoA, NADH and carbon dioxide. Now, as we have learned that 1 molecule of glucose is giving 2 molecules of pyruvate, this 2 molecules of pyruvate, when it is getting converted, we are getting 2 molecules of acetyl CoA, 2 molecules of NADH and 2 molecules of carbon dioxide. Now, this is an irreversible process. It means acetyl CoA cannot be converted back to pyruvate, hence fat cannot be converted to carbohydrate. Now, if we see the mechanism of action of pyruvate dehydrogenase complex, then we can find that this pyruvate dehydrogenase, if we are symbolizing it as E1, it is playing a very significant role. Dihydrolipoyl transacetylase, if we are considering this as E2, and dihydrolipoyl dehydrogenase, if we are symbolizing as E3, we will find that this main core, what I have already discussed with you, that pyruvate dehydrogenase core moieties, then we can find that this E1, E2 and E3 are the 3 components which are participating in this reaction. Besides that, there are 5 coenzymes which are actively involved in this reaction, and those coenzymes are thymine pyrophosphate, which is designated as TPP, lipoate, coenzyme A, FAD and NAD. These are the 5 coenzymes which are involved in conversion of pyruvate to acetyl CoA. Now, if I am showing you these are the 3 moieties E1, E2 and E3. E1 is pyruvate dehydrogenase, E2 is dihydrolipoyl transacetylase and E3 is dihydrolipoyl dehydrogenase. These 3 enzymes are participating with the 5 coenzymes and they are converting pyruvate to acetyl-CoA. This is the structure of this TPP. This is the structure of lipoic acid. Here this sulfur bonds are there, and when we are coming to this first reaction pyruvate dehydrogenase, this introduction of pyruvate onto TPP with the enzyme E1 is very significant. Now, this TPP is the cofactor, and this enzyme, when it is the enzyme pyruvate dehydrogenase, when it is attached to this TPP, it releases one molecule of carbon dioxide and hydroxyethyl TPP complex is formed. Now, if we see this reaction, this TPP is, this is the first reaction. E1 is the enzyme to which this TPP is attached as the prosthetic group, and pyruvate is coming in contact with this complex, and one molecule of carbon dioxide is released from this reaction, and as a result we are getting alpha hydroxyethylamine phosphate. So, this is the end product. Now, when this first reaction is over, then coming to the second step, what we are getting, the end product of the first reaction is coming in contact with the second enzyme E2 along with the lipoyl group, and here this particular enzyme dihydrolipoyl transacetylase, this enzyme is forming, it is transferring, transfer of acetyl group to the sulfur atom is taking place with the help of dihydrolipoyl transacetylase enzyme. And when this particular reaction is going on, then we are getting the end product. This is the end product of this reaction, and we are getting this the reaction where this product where coenzyme SH is coming in contact with this, and when this coenzyme SH is coming, the acetyl group is enzymatically transferred from this lipoyl group of dihydrolipoic acetic acid to the thiol group of coenzyme A, and this way the transfer of this acetyl group is taking place, and this enzyme is participating in this role. When this product is formed, then the E3 enzyme, which has one FAD as a cofactor, is coming and playing a significant role, where transfer of hydrogen from FAD to FADH2 is taking place, and it forms this particular E3 FAD complex, and this E3 FAD complex, in presence of this E3 FADH2, when it is just reacting, it forms this in presence of another cofactor NAD, it forms E3 FAD and NADH and H, and with the release of acetyl-CoA, and in this way we are getting this acetyl-CoA in this particular reaction. This E2 is then transfer this acetyl to CoA and form acetyl-CoA, which is the product which leaves from this complex, and this acetyl-CoA, as I have told you, this product, and this E3 uses the bound coenzyme FAD to oxidize lipoamide back to the disulfide and generating FADH2. FADH2 is recovered from FAD is recovered from FADH2 via reducing NAD to NADH, and NADH is generated as a byproduct with the completion of the reaction. That means, here this coenzyme A is produced, and here we have seen that NADH is also produced after the completion of this particular cycle. Now, this particular reaction is regulated, and if we see the regulation of pyruvate dehydrogenase, then we can find that it is irreversible reaction must tightly be controlled, and this control is taken place by 3 ways. Allosteric inhibition, that means it is inhibited by the end product of acetyl-CoA and NADH. So, what is there? That being the allosteric enzyme, the switching on switching off process is there. If the concentration is very high, acetyl-CoA and NADH, it switch off the production of acetyl-CoA and NADH. It is also inhibited by ATP. The allosteric activation of AMP is also there. That means, the ratio of ATP and AMP is also playing a significant role as far as this regulation of pyruvate dehydrogenase is there. That means, when this ATP concentration is very less, then AMP is boosting, it is the stimulating the reaction, and reaction is taking place. And the third way of controlling this is the phosphorylation and dephosphorylation of the E1, that fast moiety, that E1 subunit of pyruvate dehydrogenase. Now, what is happening inside this cell? This E1, when it is phosphorylated, this phosphate is getting attached, it becomes inactive, and when with the help of the enzyme phosphatase is taking place, then it becomes E1 hydroxyl group is there, and it is active, and it converts this process, and ATP is being utilized and ADP is produced. So, when in the cell ADP concentration is very high, then what is happening? When ADP concentration is very high, pyruvate is very high, it inhibits this particular enzyme, and total reaction is getting stopped. And this is the switching on switching off process, which is controlled by this allosteric function of this enzyme. Now, this kinase enzyme is activating here, that is the transfer of phosphate group, that kinase mainly transferring the phosphate group of this ATP triphosphate, and it becomes diphosphate, and this enzyme is phosphorylated, and when it is dephosphorylated, then it is once again active, and in this way the active and inactive forms are going on inside the cell depending upon the need and requirement of this particular product in the cell. Now, if we sum up this pyruvate dehydrogenase complex, then we can tell this particular thing what I have already mentioned, as this pyruvate dehydrogenase complex consists of 3 enzymes, that is pyruvate dehydrogenase E1, dihydrolipoyl transacetylase E2, and dihydrolipoyl dehydrogenase, that is E3. It has got 5 cofactor coenzyme ASH, NAD, lipoamide, TPP and FAD. Coenzyme ASH and NAD participate in the stoichiometrically in the reaction, and the other 3 cofactors have catalytic function of the particular enzymes concerned. TPP is a cofactor for E1, lipoamide and coenzyme SH are the cofactor for E2, and FAD and NAD are the cofactor for the submoiety of E3. The pyruvate dehydrogenase reaction is irreversible, and overall reaction is this pyruvic acid, coenzyme ASH and NAD gives rise to acetyl-CoA, carbon dioxide and NADH. This is called as activation of pyruvate. In the TCA cycle, intermediates are activated by formation of high energy thioester bond. One carbon of pyruvate is oxidized to carbon dioxide and one NADH is formed by the end of this reaction. Now, if we see the regulation, then pyruvate dehydrogenase step is irreversible. As a result, animals are not able to synthesize glucose from acetyl-CoA or fat. Pyruvate dehydrogenase is regulated allosterically and by the covalent phosphorylation, as we have already discussed this. Pyruvate dehydrogenase is inhibited by acetyl-CoA, that enzyme is dihydrolipoyl transacetylase, and NADH, that is E3 moieties of dihydrolipoyl dehydrogenase, being allosteric in nature. Covalent phosphorylation of pyruvate dehydrogenase turns off its activity and dephosphorylation results in the activation. High ATP, NADH and acetyl-CoA stimulate the pyruvate dehydrogenase kinase. Insulin, NAD and ADP stimulate pyruvate dehydrogenase phosphatase to act. Now, if we see the TCA cycle and its overview, and then if we enter to the actual TCA cycle, then it will be a little bit it will be easy for understanding. It is the cyclic pathway. All intermediates are derivative of citric acid, a tricarboxylic acid. This is the 8 steps process, that means glycolysis is a 10 steps process, but TCA cycle is the 8 steps process. All except 3 are reversible in nature. Acetyl-CoA, the 2 carbon molecule enters the cycle by condensation with the 4 carbon oxaloacetate, and each of these 2 carbon of acetyl-CoA are oxidized and remove the carbon dioxide in 2 separate reactions. Oxaloacetate is regenerated and reused in this process. 3 molecules of NAD and 3 molecules of NADH and H plus are produced. 1 molecule of FAD is required and 1 molecule of FADH2 is produced. 1 molecule of GDP is phosphorylated to 1 molecule of GTP, which is otherwise known as substrate level phosphorylation. And in case of glycolytic process, I have also mentioned about this substrate level phosphorylation, and here also we will see that substrate level phosphorylation reactions. Now, coming to the actual steps. Now, acetyl-CoA is formed. Now, acetyl-CoA is entered to the mitochondria. In the mitochondria, oxaloacetate is present. Now, inside this mitochondria, when this condensation reaction is going on in presence of the enzyme citrate synthase, then this synthesis of these 2 different molecules, one is 2 carbon, another is 4 carbon, is taking place, resulting in the formation of citric acid. 1 molecule of water is needed and coenzyme SH is coming out as a byproduct of this reaction. Now, citrate is an intermediate in TCA cycle. After the pyruvate dehydrogenase complex form acetyl-CoA from pyruvate, a 5 cofactor thymine pyrophosphate TPP lipoamide FAD NAD coenzyme A citrate synthase catalyzes the condensation of oxaloacetate with acetyl-CoA to form citrate. Now, here you see acetyl-CoA and here oxaloacetate. You see 1, 2, 3, 4 carbon moieties oxaloacetate. In presence of citrate synthase, it is producing some intermediate product called S-citryl CoA intermediate, and after a proton is removed from this CH3 group of acetyl CoA, the negatively charged CH2 forms a bond to the carbonyl carbon of the oxaloacetate, and the subsequent loss by the hydrolysis of the coenzyme A drives the reaction forward, and it results in the formation of citrate. And this is the first step of citric acid cycle. In the second step, what is there? Citrate is converted to aconitate and isocitrate. Now, aconitase is the enzyme that catalyzes the stereospecific isomerization reaction of citrate to isocitrate via cis aconitate in the tricarboxylic acid cycle. Now, if we see here, this is the isomerization reaction in which the water is removed first. One molecule of water is removed first, followed by the addition of water. It moves the hydroxyl group. Now, this hydroxyl group which is there from one carbon to the another neighboring carbon, and it forms the product which is called isocitrate. This is the second step. In the third step of TCA cycle, isocitrate is converted to alpha ketoglutarate, and here in this particular reaction what we can see that product is that isocitrate to alpha ketoglutarate, and it release one molecule of carbon dioxide. When one molecule of isocitrate is utilized, it produce one molecule of carbon dioxide. Here you see this isocitrate dehydrogenase enzyme is needed, is actively participating in the catalytic activities, and here one molecule of NAD is converted to NADH. That means, we can also get one molecule of NADH in this particular reaction. That means, as a result what we are getting, this is that decarboxylation reaction is taking place, that oxidative decarboxylation of isocitrate producing alpha ketoglutarate, and carbon dioxide is being released in this process, while NAD is getting converted to NADH, and this is a two-step reaction which involves the oxidation of isocitrate, a secondary alcohol, to oxalosuccinate, a ketone, followed by the decarboxylation of carboxyl group beta to ketone, forming alpha ketoglutarate. So, this is the isocitrate. Here isocitrate dehydrogenase NAD to NADH is produced, oxalosuccinate intermediate is there, and here one molecule of carbon dioxide is being released, and we are getting alpha ketoglutarate. The first of the 4 oxidation steps in this cycle, the carbon carrying the hydroxyl group is converted to carbonyl group, and the immediate product is unstable, losing a molecule of carbon dioxide while still bound to the enzyme, and this is the third step of TCA cycle. In the fourth step, you see alpha ketoglutarate is converted to succinyl CoA. Now, this reaction mainly takes place in 3 steps. Now, decarboxylation of alpha ketoglutarate, reduction of NAD to NADH and subsequent transfer of coenzyme A, which forms the end product of succinyl CoA. Now, that means here alpha ketoglutarate dehydrogenase enzyme is here. One molecule of NAD is also, that means in step 4, that is alpha ketoglutarate to succinyl CoA. Succinyl CoA. And here also we can find that one molecule of carbon dioxide is released and one molecule of NADH is released and coenzyme SH is required to form succinyl CoA. Now, if we see the reaction, then we can find that this is alpha ketoglutarate, this is 1, 2, 3, 4 and 5 carbon moieties. Here, when one molecule of carbon dioxide is going and coenzyme is coming and getting bound with this particular succinyl and form this succinyl CoA, this becomes 1, 2, 3, 4 carbon compound. So, this is the reaction. The alpha ketoglutarate dehydrogenase complex closely resemble to the large complex that convert pyruvate to acetyl CoA. It catalyzes the oxidation that produce NADH, CO2 and high energy thioester bond to coenzyme A. So, that means thioester bond is also produced with this, which is linked with this coenzyme A. In the 5th step of this TCA cycle, succinyl CoA is converted to succinate and coenzyme A is coming out of this process. And here in this process, in step 5, this succinyl CoA to succinate, here one molecule of GTP is produced and this GTP in case of bacteria and plant, it is in the form of ATP. Now, succinyl CoA synthetase enzyme is releasing that coenzyme A and it forms this succinic acid. Succinyl coenzyme A synthetase, that it is a thiokinase enzyme, catalyzes the formation of succinate and coenzyme A 4 carbon metabolite that form a coenzyme A. This is the 4 carbon compound and coenzyme A is getting released and this is succinic acid is produced, and here one molecule of GTP is produced and this is otherwise GDP is converted to GTP in the presence of inorganic phosphate. One molecule of water is also needed to carry out this reaction. A phosphate molecule from solution displace the coenzyme A, forming a high energy phosphate linked succinate, and this is also the example of substrate level phosphorylation what I have already mentioned you in case of glycolytic process. And this GTP which is there in the higher in animal system and it is in the form of ATP in case of bacteria and plant. In the 6th step, succinate is converted to fumarate. That means, in 6th step, succinate is converted to fumarate. And here we are getting one molecule of FADH2, one FAD molecule. This particular succinate dehydrogenase is the enzyme which is not present in the mitochondrial matrix, but this is a membrane bound enzyme which is found in the inner membrane of mitochondria, and here one FAD molecule is getting converted to FADH2. Now, here if we see the reaction, you see this is the succinate, and in presence of FAD it is forming the fumarate. This is the third oxidation step in this cycle, where FAD removes 2 hydrogen from and form the succinate. So, here this hydrogen is also produced in this reaction. 2 molecules of hydrogens are produced in this reaction. Now, when fumarate in the next step, that step 7, that fumarate is getting converted to malate, and in the presence of enzyme fumarase, and here one molecule of oxygen water is needed and it forms the malate. You see the addition of water to the fumarate places the hydroxyl group next to the carbonyl carbon, and it forms the malate. And when this malate is converting taking part, and it converted to oxaloacetate, the first product of TCA cycle in presence of the enzyme malate dehydrogenase. That means, in step 8, that malate is converted to oxaloacetate, and here we can get one molecule of NADH and oxaloacetate. So, if we see this reaction, then we can find that, you see, in presence of malate dehydrogenase, in the last 4 oxidation steps in this cycle, the carbon carrying the hydroxyl group is converted to the carbonyl group, regenerating the oxaloacetate needed for the first step of this reaction, and this is once again the 4 carbon compound, and in this way leads. Now, if we see the total TCA cycle, then we can find that, you see, acetyl CoA is coming, and it is coming in contact with this oxaloacetate. Condensation reactions are going on, and it forms the citrate, the 6 carbon compound, then citrate to isocitrate, isocitrate to alpha ketoglutarate, alpha ketoglutarate to succinyl CoA, followed by succinate, fumarate, malate and oxaloacetate, and in this way the total cycle gets completed. And when this cycle is getting completed, we are calling that, yes, this cycle is continuing and cellular respiration is going on inside the mitochondria of the cell. Now, if we see the summary of this particular reaction, then we can find that introduction of 2 carbon atom and their loss yielding 2 NADH and a GTP or ATP equivalent to ATP, partial oxidation of succinate to oxaloacetate, another NADH is produced as well as a reduced FADH2, oxaloacetate is regenerated for the next cycle, and if we see the overall reaction, then we can write these reactions like this: acetyl CoA and 3 molecules of NAD, because we can see that this is 1, this is 2 and this is 3. That means 3 molecules of NAD is needed, 1 molecule of FAD, 1 molecule of FAD, 1 molecule of GDP and PI, and in presence of 2 molecules of water gives rise to 2 molecules of carbon dioxide. See, 1 molecule and another molecule, this is 1 carbon dioxide and this is another molecule of carbon dioxide. So, it results 2 molecules of carbon dioxide, 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of GTP, 2 H plus, 2 H plus and 1 coenzyme A, which is produced in this 1 molecule of pyruvate. And when 1 glucose is producing 2 molecules of pyruvate, then this entire thing is getting double. One high energy compound is made, 4 pairs of electrons are made available to the respiratory chain of oxidative phosphorylation. These are used to generate most of these ATP which are needed. And if we see the ATP from the citric acid cycle, we can find that ATP from citric acid cycle, that oxidation of 2 isocitrate, the 2 molecules of NADH is there. That means 1 molecule of NADH is equivalent to 3 molecules of ATP, that means it is 6 molecule of ATP equivalent. Oxidation of 2 alpha ketoglutarate is also giving that 6 molecules of ATP. 2 direct substrate level phosphorylation is giving 2 molecules of ATP. Oxidation of 2 succinate is giving 2 molecules of FADH2 and 1 molecule of FADH2 is equivalent to 2 molecules of ATP. That means it is 4 molecules of ATP because 2 molecules of FADH2 is there, and oxidation of 2 malate is also producing 2 molecules of NADH and it is giving 6 molecules of ATP, and altogether we can, if we sum up, we are getting 24 molecules of ATP, and if we summarize the reaction, 2 molecules of S-acetyl CoA gives rise to 4 molecules of carbon dioxide, 2 molecules of water and 24 molecules of ATP. And if we see the overall ATP generation in this process starting from glycolysis to TCA cycle, then we can find that, as we have already learned, that 8 molecules of ATP is produced in glycolytic pathway, and we have also seen that when pyruvate dehydrogenase is participating in conversion of pyruvate to acetyl CoA, one pyruvate is giving 1 NADH and 2 pyruvate is releasing 2 molecules of NADH equivalent to 6 molecules of ATP. TCA cycle, in TCA cycle we have already seen that 24 ATP molecules are formed. So, if we overall sum up that from glucose to the completion of TCA cycle, we can find that 38 ATP molecules are formed. Now, as I have already mentioned that it is the regulation of citric acid cycle. Now, pyruvate dehydrogenase is the enzyme, citrate synthase is the enzyme, isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase are the enzymes which are regulating, which are playing a significant role on the regulation of citric acid. Now, when we are talking about the regulation, we can find that the intramitochondrial NAD/NADH ratio, high oxygen level results in the increased ratio and low level of and low levels, a decreased ratio, a measure of oxygen availability are playing a major regulatory role in citric acid cycle. Now, coming to this anabolic role of TCA cycle, we can find that the intermediates of TCA cycle serves as the precursors for biosynthesis of biomolecules. Many amino acids are synthesized starting with transamination of alpha ketoglutarate. Porphyrin and heme are synthesized from succinyl CoA. Oxaloacetate is another alpha keto acid and its transamination leads to aspartate and other amino acid biosynthesis. Oxaloacetate is also the precursor for purine and pyrimidine via aspartate. and fatty acid and sterols are also synthesized from citrate, and these are the anabolic role of TCA cycle. If we see the anaplerotic reactions, we will find that since the TCA cycle intermediates are used for anabolism, their concentration varies according to the need of a particular cell. Reactions that replenish the TCA cycle intermediates are also called as anaplerotic reactions. Oxaloacetate can be considered as the primary substrate for TCA cycle. It is replenished from pyruvate by gluconeogenic enzyme pyruvate carboxylase. Now, pyruvate plus carbon dioxide, ATP and water gives rise to oxaloacetate, ADP and PI. Pyruvate carboxylase is activated in the presence of acetyl-CoA. Pyruvate can also replenish malate, and in this way the anaplerotic reactions are going on in TCA cycle. And I have tried to give you the overview of this catabolic process, as I have told you earlier that the reactions are of two types. One is the anabolic reactions, another the catabolic reaction. Now, we are discussing the respiratory process, which is a catabolic reaction, and what I am discussing that from glycolytic pathway, how TCA cycle it starts and how pyruvate is converted to acetyl-CoA, and acetyl-CoA entered to this mitochondria and it completes the cycle, and this cycle continues in the living system, and this way this TCA cycle is taking place in mitochondria of any cell. So, thank you very much, students. And in the next class, I will be talking about this the electrons which are produced, which are there, how they are being carried out in this particular reaction to complete the cellular respiration. Thank you very much.